Did you know that the fats you eat can actually influence your genes, not just in a vague, indirect way, but in a direct, measurable way that impacts how your body stores and uses energy? That's wild. I think most of us think of fats as either fuel or something that gets stored in our bodies, but you're saying they're more like messengers? Exactly. Today, we're talking about how fatty acids regulate gene expression. It's a fascinating process that connects diet, metabolism, and even chronic diseases like diabetes and heart disease. Okay, so let's start with the basics. What does it mean for a fatty acid to regulate gene expression? At its core, it means that the fats we consume can turn certain genes on or off. These aren't just any genes, they're the ones that control how our bodies process and store energy. This mechanism is thought to be one of the earliest ways organisms adapted to their environments. It's not just about energy, though, it's also tied to diseases like diabetes, cancer, and cardiovascular issues. So, fatty acids are more than just a source of energy or something that gets stored. They're actually active players in how our cells function. How do they do that? The article we're discussing outlines two main mechanisms. First, there's the direct modification of gene transcription. Think of it like flipping a switch to turn a gene on or off. Second, fatty acids can change the composition of fats in cell membranes, which affects how cells communicate and respond to hormones. So they're influencing gene expression both directly and indirectly. That's already pretty fascinating. But the article also connects this to diabetes. How does dietary fat tie into that? Historically, diabetes was seen as a problem with glucose metabolism, but now we know that type 2 diabetes, or non-insulin dependent diabetes mellitus, is also a disorder of lipid metabolism. High-fat diets, in particular, have been shown to cause insulin resistance. That's interesting. I think most people associate diabetes more with carbs than fats. How does a high-fat diet lead to insulin resistance? A high-fat diet disrupts insulin signaling and receptor binding. This leads to a cascade of problems like increased glucose production by the liver and reduced glucose utilization by both the liver and skeletal muscles. Adipose tissue or body fat becomes less efficient at forming glycerides, and skeletal muscles have reduced glucose oxidation. That sounds like a chain reaction of issues. But what's happening on a cellular level? Is it just that less insulin is binding to receptors? It's more than that. Studies show that high-fat diets can reduce insulin binding by as much as 50 to 75%. This reduction in binding is accompanied by decreased glucose uptake and oxidation in cells. Additionally, high-fat diets suppress the activity of an enzyme called pyruvate dehydrogenase, which is critical for glucose metabolism. On top of that, they lower levels of a glucose transporter protein called GLUT4, which is essential for insulin-stimulated glucose uptake. So, high-fat diets are clearly a factor in insulin resistance. But what about insulin secretion itself? Does dietary fat affect how the pancreas releases insulin? It does. Dietary fat can interfere with glucose-stimulated insulin release from pancreatic beta cells. Normally, glucose acts as a signal for the pancreas to release insulin, but prolonged exposure to free fatty acids can suppress this process. Specifically, it inhibits the transcription of acetyl coenzyme A carboxylase, an enzyme that plays a role in insulin release. That's interesting. So fatty acids can actually inhibit insulin secretion over time. But does this happen with all types of fats, or are some worse than others? That's where things get nuanced. Not all fatty acids have the same effects. There's something researchers call the fat paradox. Saturated fats, monounsaturated fats, and polyunsaturated fats each influence glucose and lipid metabolism differently. For instance, diets high in N6 polyunsaturated fatty acids can improve insulin binding and glucose transport compared to diets high in saturated fats. However, even high N6 fat intake can impair glucose metabolism under certain conditions. So it's not as simple as labeling fats as good or bad, but what's the key difference between these types of fats? Are there specific ones we should aim to include in our diets? The article highlights that replacing some N6 fatty acids with long chain N3 fatty acids, like those found in fish oil, can significantly improve glucose metabolism. In fact, these N3 fatty acids can enhance glucose disposal in muscle and adipose tissue almost to the level seen with a high carbohydrate diet. So, fish oil seems to be the star here. It helps with glucose uptake in muscles and fat tissue. But does it have the same effect on the liver? Not quite. While N3 fatty acids improve glucose utilization in muscle and adipose tissue, they actually suppress the liver's use of glucose for fatty acid and triglyceride production. Instead, they promote fatty acid oxidation and ketone body synthesis in the liver. Essentially, they act as fuel partitioners, redirecting glucose and fatty acids toward different metabolic pathways. That's fascinating. It's like these fatty acids are orchestrating a coordinated effort across various tissues. But how does all of this tie back to gene expression? How do fatty acids actually influence which genes are active? That's the crux of the matter. Polyunsaturated fatty acids, particularly those rich in 20 and 22 carbon N3 fatty acids, reduce hepatic triglyceride output by suppressing the transcription of genes involved in fatty acid and triglyceride synthesis. They also increase the transcription of genes involved in fatty acid oxidation. So it's not just about changing enzyme activity, it's about reducing the production of the enzymes themselves. How quickly does this happen? Very quickly. For instance, replacing a fat-like triolein with menhaden oil can suppress the transcription of genes like fatty acid synthase and pyruvate kinase in less than 3 hours. This rapid response suggests that polyunsaturated fatty acids directly modulate gene transcription, bypassing other mechanisms like changes in membrane composition or hormone release. That's incredibly fast, but what's the molecular mechanism behind this? How do these fatty acids actually get into the nucleus to affect gene transcription? The article discusses the role of transcription factors called peroxisome proliferator activated receptors, or PPARs. These are proteins that can be activated by ligands, including fatty acids. Once activated, PPARs influence the expression of genes involved in lipid and glucose metabolism. I've heard of PPARs before, so are fatty acids directly binding to these receptors and changing gene expression? It's a bit more complicated than that. While PPARs are certainly involved, the article points out that saturated, monounsaturated, and polyunsaturated fatty acids all have similar potency as PPAR activators. This doesn't fully explain the unique effects of N6 and N3 fatty acids on gene transcription. There's evidence suggesting that some of these effects occur through PPAR-independent pathways. It sounds like there's still a lot to learn about these mechanisms, but what's the big takeaway here? The key takeaway is that dietary polyunsaturated fatty acids, particularly N6 and N3 fatty acids, play a unique role in regulating fatty acid biosynthesis and oxidation by modulating the expression of genes coding for key metabolic enzymes. These changes influence how our bodies metabolize glucose and fats, which has profound implications for diseases like diabetes and potentially other metabolic disorders. It's amazing how much influence our diet can have, not just on our immediate health, but on the very genes that control how our bodies function. Absolutely. This field is still evolving, but understanding how fatty acids regulate gene expression is crucial for developing better strategies to prevent and treat metabolic diseases. Thanks for breaking this down. It's a lot to take in, but it's such an important topic. You're welcome, and thanks to everyone listening. There's always more to explore in the world of science and health, so stay curious. Goodbye. Goodbye.